This video describes how to culture human pluripotent stem cells on Biolaminin 521. Biolaminin 521 is a human recombinant laminin cell culture substrate produced and distributed by Biolamina. The day before splitting the cells, prepare a Biolaminin 521 coated tissue culture plate according to the cell cultureware coating instructions. If you do not have plates prepared, you can quickly coat a fresh plate at 37 degrees Celsius for 2 hours. Start by aliquoting sufficient amounts of 1 times DPBS, cell dissociation reagent and medium needed for the protocol, and pre-warm to 37 degrees Celsius. Recommended volumes specific for different cultureware formats can be found in the written protocol. The protocol can easily be made totally defined and xeno-free with your choice of culture medium, since the Biolaminin 521 matrix works well in combination with most commercial media brands. You can also use a cell dissociation reagent of choice, since both enzymes and EDTA-based dissociation can be used. Carefully aspirate the Biolaminin 521 solution from the coated wells without disturbing the coated surface. Immediately add fresh culture medium to each well. Do not allow the coated surface to dry, as this will inactivate the coating. Place the plate in an incubator to let the medium equilibrate. When culturing the human pluripotent stem cells on Biolaminin 521, the cells are ready to passage between 60 to 90% confluency. The cells should have formed a homogeneous monolayer, and the culture should not contain any differentiated areas. Bring out the plate with cells that are ready to be passaged, together with a pre-warm 1 times DPBS and the dissociation agent. Aspirate the medium from the cells and wash the cells gently with 1 times DPBS. DPBS without calcium and magnesium should be used, since divalent cations may have a negative effect on some dissociation enzymes. Aspirate the DPBS solution and add the cell dissociation agent. Incubate at 37 degrees Celsius for 3 to 5 minutes or until the cells start to round up. The incubation time is cell line dependent and differs depending on the dissociation agent used. A more confluent culture and the use of a high coating concentration could make the cells attach more tightly to the surface, and a prolonged treatment time with a dissociation agent may be required. Gently aspirate the cell dissociation solution. Add pre-warmed fresh medium and gently rinse the well to detach the cells. The cells should detach easily, and no more than 2 to 3 washes should be required. Use a microscope to verify that the cells are properly dissociated. When using Biolaminin 521, we recommend passaging pluripotent stem cells as single cells or as small aggregates. When using EDTA, try to achieve as small cell aggregates as possible without using too much mechanical force. The mechanical force applied should be minimised, since extensive pipetting may result in low cell viability. Rather, increase the incubation time with a dissociation reagent to minimise the mechanical force needed. Transfer the cell suspension to a 15 ml tube and centrifuge at 100 times G for 4 minutes at room temperature. If you're using EDTA as a dissociation agent, you can skip the centrifugation step. Aspirate and discard the supernatant and resuspend the pellet in fresh, pre-warmed culture medium. Count the cells and calculate the cell suspension volume needed for seeding. Bring out the fresh Biolaminin 521 coated tissue culture plate containing fresh, pre-warmed culture medium and transfer the desired volume of cell suspension to each well. The cells rapidly attach to the Biolaminin substrate, so immediately rock the plate to get an even cell distribution. Human pluripotent stem cells can routinely be cultured as single cells on Biolaminin 521 without the need for ROCK inhibitor, but if you are transitioning your cells to Biolaminin 521 from feeders or another feeder-free matrix, we recommend adding ROCK inhibitor to a final concentration of 10 micromolar for the first few passages. Once adapted to the Biolaminin matrix, pluripotent stem cells can routinely be cultured as single cells without the need for ROCK inhibitor. Place the plate into the incubator and incubate at 37 degrees Celsius and 5% carbon dioxide at high humidity. Perform a complete medium change 24 to 48 hours after passaging, and then feed the cells on a daily basis until the next passage. If you experience an uneven cell growth, it's often a coating-related issue and could be caused by the following. 1. that too low a coating concentration has been used, causing the cells to have difficulty in migration and reaching confluence. The cells will form clumps rather than monolayer colonies in combination with more floating cells. Slightly increase the coating concentration until it is high enough to support an evenly distributed monolayer growth. 2. Uneven cell growth could also be caused by bad coating coverage. Ensure that the entire surface is covered by the coating solution when preparing fresh plates. Also, do not let the plate dry, as this will inactivate the coating. Too long incubation times or long storage in the fridge could cause the plate to dry out, especially at the centre of the plate. For more information on how to culture human pluripotent stem cells on Biolaminin 521 and other procedures, please visit our website at www.biolamina.com.